My name is Aline Souza. I am a master's student rehabilitation science program at Universidade 9 de Julho in Brazil. I am pleased to present the study title, Evaluation of the Blue LED on the L929 fibroblast cells viability. Fibroblasts are key cells that play a crucial role in maintaining the structural integrity of muscles, as well as being responsible for the deposition and renewal of the extracellular matrix. During the tissue repair process, fibroblasts, initially in a quiescent state, undergo phenotypic differentiation, adopting a proliferative and contractile phenotype. This process plays a fundamental role in promotion wound repair, activating immune responses, and differentiation in other cell types. And here I present some authors who assessed photobiomodulation in other cells and their findings, such as Ramos et al. Employing red laser irradiation for their assessment of tissue repair. Improvements were observed in epidermal organization and deposition collagen fibers. Brito et al. In an in vitro experiment, it was observed that the red laser had the capacity to modulate the excitation of acute inflammation with 24 hours time frame, leading to a reduction in pro-inflammatory chemokines. And Malavasi et al. Photobiomodulation demonstrated the capacity to decrease the concentrations of pro-inflammatory proteins and markers, crucial for facilitation myoblast differentiation during the process of muscle tissue repair. Viability is a fundamental factor to be assessed for estimating survival, apoptosis, and responses to external stimuli, and in this case the impact of blue LED on fibroblastic cells. Recent studies have shown benefits in proliferation, differentiation, influence at mitochondrial and reticular activity when utilizing photobiomodulation with low-level laser or LED. However, the ideal dosimetric parameters are not yet comprehensible. Taking into account the factors that PBM exerts on various cells type and the importance of the function the fibroblasts perform in different tissue and the tissue repair process, and given the limited availability of study that illustrated ideal dosimetric parameters in relation to blue LED radiation. This study opens the door for applicability in the clinical context in the future. Therefore, this study aims to assess different dosimetric parameters of blue LED light on the L929 fibroblast cell viability. The experimental groups were divided into control group, L929 and culture medium DMM, plus 10% fetal bovine serum. L929 group in DMM with blue LED irradiation at 4, 6, and 8 joules. The choice of parameters for treatment with blue LED was based on previously published study of Alt's research group. The L929 fibroblast would culture NDMM with 10% FPS. Remove it using trypsin and DTA, centrifuged, and counted. Separated into tubs to form the experimental groups, and were irradiated with blue LED using the respective parameters. The cells were then incubated in 96 wells plate to a year for 24 and 48 hours. After each time point, GMM was collected and MTG was added, followed by a 3 hours incubation period. Following incubation, isopropanol was added to form parmesan crystals, which were then subjected to absorbance readings and analysis. Data generated from the MTT assay for evaluation of cell viability were analyzed using the GraphPad Prism 8 program. The data were expressed as mean and the standard error of the mean. Parametric data were compared among groups using ANOVA 2K. For the results, present graph A for the 24 hours analysis and graph B for the 14 hours analysis, showing the percentage of cell viability. The control groups are represented in orange, followed by the 4 joules in pink, 6 joules in green, and 8 joules blue-greens group. It is noticeable that there was an increase in cell viability at 14 hours in the 8 group compared to the 6 group. However, there was no statistical difference at 24 hours. In conclusion, blue LED radiation applied at 8 joules was able to time-dependently simulate the viability of L929 cells. I would like to thank the organizers of the Congress, my institution, the Universidade 9 de Julho, and the funding agencies CAPES and FAPESP for their support. I also wish to thank the professors and my research group. Finally, I express my acknowledgement to all those present for their interest in